Good morning, everyone. I am Dr. Chandrasekhar, Medical Director of the Resplice Autism Research Institute. Today, we are going to talk about solutions to the autism at individual level and community level. So, from our institution, we are working on certain projects which actually will have huge impact in the society in the future. One of the major project is gut microbiome transplant. We believe that gut microbiome transplant will improve quality of life of the autistic children significantly. And another project we're working on is we are working on a public health policy called NSTEL, inclusion of NSTEL 16 into the antenatal program, so that will reduce the autism incidence in the future. So before explaining the projects completely, let us start with the neurobiology of autism, molecular biology of autism, then why these gut microbiome transplant and NSTL 16 and other things are relevant for the autism. Autism is a big social problem now, but it is very much underestimated in the society, in the public health sector or anywhere. We can actually see a lot of increase in the incidence in last 4 decades. So it was 1 in 10,000 in 1980s, and now by 2020 reached up to 1 in 54. You can make a note of something from the history. So Roman Empire got collapsed due to lead poisoning. And the consequence was the low birth rates and mental incompetence. In today's society also, you can see the increased infertility rates and autism, intellectual disability. It's all similar to that. So we know that Roman Empire collapsed with lead poisoning. Now, similar to lead poisoning, we are consuming lots of toxic compounds. Maybe it may be pesticides, weedicides, industrial toxin, and preservatives, all these things. So many things we are consuming. This is actually the major cause for the rise in incidence of autism and infertility. In current days, the standard practice in autism is there is no established cause for autism, and there is no established treatment for autism, and the therapies like ABA, behavioral therapy, occupational therapy, speech therapy are the rehab options, and risperidone and aripiprazole are the FDA-approved drugs that help to some extent. This is the standard practice in the autism, and this is a status quo situation till now, and somebody needs to work beyond that to get some impactful research onto the track. So to understand autism better, we need to look into the neuroembryology aspects. So today I will present you from the point of fertilization. At what stages autism starts occurring, and what are the aberrant pathways that occur in autistic brain while neurodevelopment. So when after fertilization, a single cell zygote is formed. That single cell zygote divides into 2, then 4, and then form a ball of cells called morula. You can see here. So this morula develops a cavity, and this stage is called blastocyst. And in this blastocyst, some of the cells become embryoblast. These embryoblast cells are the ones which develop into the fetus. So this is like this is a 2-dimensional structure. You can see this is actually a hollow sphere kind of thing with a mass of cells called embryoblast here. So in this particular this image, you can see this trophoblast has developed another cavity above the embryoblast. Then this embryoblast becomes like a bilaminar disc. So this disc is the actually like this disc is going to become the entire human body. So it has 2 layers of the cells. So upper layer is called epiblast, lower level is called hypoblast. So if you look at from the top end of this disc, you can see this is the upper surface you are seeing is a epiblast, The lower surface is the hypoblast. On the epiblast, so a knot-like structure here forms like primitive node. We call it as primitive node. From primitive node, in the midline of the disc, a line or a groove develops. This is called primitive streak. The cells here degenerate and forms a groove kind of thing, and a, pronotochordal cells from this primitive node travel in the midline and form a notochord. This notochord is going to become the spine in the future. So this is the cross section of that bilaminar disc. So after this is the primitive streak that I was talking about. So after this primitive streak is formed, cells from the epiblast become mesodermal cells and then travel in between these two layers, epiblast and hypoblast, and form a third layer. So then after formation of the third layer, here you can see, so after formation of the third layer, the epiblast cells become the ectoderm, hypoblast cells become the endoderm, and middle layer becomes the mesoderm. This is a trilaminar disc formation, and you can see the notochord here. This notochord starts developing, starts producing certain growth factors. So this is the notochord. So this notochord starts producing certain growth factors that convert this ectoderm cells into neuroectoderm. So this is the neuroectoderm. Once this neuroectoderm is developed, then this particular neuroectoderm is called neural plate. This neural plate slowly invaginates. So this is if you see this upper layer, this is the entire ectoderm. This is the neural plate, which is neuroectoderm. This invaginates like this and forms a tubular structure called, this is a neural tube. So here you can see, so this is a tubular structure that is formed in the neural plate. So it has two openings. One is anterior neuropore and posterior neuropore. So if the anterior neuropore fails to close, then though in those conditions, conditions like anencephaly develops, and which is incompatible for life, and these anencephaly fetuses die immediately after birth. And many other conditions like spina bifida, many conditions also there that can go wrong at this stage. So once this anterior neuropore and posterior neuropore get closed, this neural tube develops three vesicular structures like this. The most anterior part is like it is called rostral end. Rostral end is converted into the prosencephaly. Middle one converted into mesencephalon and rhombencephalon. So this prosencephalon is further divided into the telencephalon. You can see this structure, Mickey Mouse kind of structure. So this is the telencephalon, and this is immediately before the telencephalon you can see diencephalon. This part is the one that is going to develop into the like forebrain and midbrain. So this is the lateral view. So this is the telencephalon part. This is and diencephalon. This is going to become the brain. So till this point, nothing is going wrong in autism. After this stage, there are certain mechanisms going abnormal. They are called neuronal migration, neuronal differentiation, microglial dysregulation, synaptic pruning, and dysfunctional circuitry formation, abnormal neurotransmitter formation, accelerated neurogenesis. Let's discuss about each of the problem here in the coming slides. So this is the neuronal migration. So you have seen. So I'll go back to two slides before. So this is like you can see this is the telencephalon. This is the rostral end. So this part is the cross section of the telencephalon. That like we have seen the Mickey Mouse kind of structure. No? That is this particular thing is the cross section of the telencephalon. So the innermost layer, which is close to the embryonic ventricle, this is the cavity in the neural tube, which is fluid filled. So the immediate layer of the telencephalon is called ventricular zone. This is the area. This ventricular zone is the thing that produces the neuronal stem cells. The first formed stem cells produce a ladder-like cells or creeper-like cells like this. These are called radial glial cells. The subsequent batches of the neuronal stem cells track on these rope-like cells or ladder-like cells, radial glial cells, and form six-layered structure of the cortical plate. So in the neocortex, the six-layered structure is formed. In the archicortex, only three layers are formed. In the periocortex, it is 4 to 5 layers. So this is actually driven by certain signaling molecules like renin and laminin. So this is like in this particular neuronal migration in autism, renin is produced only 40% of the quantity that is present in the neurotypical. So this renin when produced in lesser quantity, neuronal migration defects occur, and the proper cortical structure is not formed. And this is another image that can explain. This is the radial glial cells, and the neuronal precursor cells track on these radial glial cells and form six-layered structure. So after reaching the destined places, these neuronal precursor cells, the initial neuronal precursor cells will be like smaller in size, less branched, and then slowly they grow in the size, and they branch and become fully mature, and they start forming connections with the adjacent neurons. So here you can see this is these two are control neurons. These two are autistic neurons. You can see it is less branched in this particular subject, and they most of them they are smaller in size. They do not grow fully, and they do not branch properly and do not make proper connections. So this is called a neuronal differentiation. Maturation of neuronal cells into different types of neurons is differentiation. So this is quite contrast we in some cases of the postmortem brain tissue studies. So in some cases they have reported that they are more branched than the neurotypical. So in most publications, I have seen that it's less branched, but they end up in middle stages of the differentiation. Because of that, there are more number of neuronal precursor cells and more number of neuronal immature neurons, and high density is there, high number of cells are there. So in this particular study, it's completely opposite. The density is high, but size is also typically greater in children with autism. So there, I could see two different contrasting publications. So so here I need to explain about one more thing, because like when the cells are failing to form the proper lamination means formation of this 6 layers or 5 layers, 4 layers, which is called lamination. When this lamination is not happening properly and when the differentiation is not happening properly, from the cortical plate there is a signaling to the ventricular zone that the brain tissue is not properly formed. Then this ventricular zone starts producing more number of stem cells. Hence, children with autism will the brain of the autistic children will have at least 100 times more number of stem cells than the normal human being. So that stem cell, that ventricular zone is generally thickened because of the excess number of production of the neural stem cells. Once this neurons grow in size and form connections with the adjacent neurons, like this. This is a neuron, brain cells. They form connections with adjacent neurons randomly. So when they connect randomly, the tight junctions called synapses. So microglia are the macrophage-like cells, like they are like phagocytoses. They eat away these synapses. So whenever there are randomly connections are formed randomly. So microglia will identify missense immature synapses and cleave it, so that the circuitry is properly formed, the connections are properly formed. So this microglia get signaling. If the microglial signaling is affected, then they get dysregulated and they do not perform to the uh, expected level, and then they end up in more number of immature synapses and missing synapses. Because of this, they have aberrant circuitry. Uh, this is the reason why they cannot process the information properly. So gross pathology includes, because of all these issues, there are three major gross pathology we can see in the postmortem brain tissue studies. That is subependymal nodular dysplasia, heterotopia, and multifocal dysplasia. So here you can see this is the adult autistic brain autopsy studies. They have seen, like you can see, this is the ventricular zone. You can see thickened ventricular zone area because the brain is not properly developed. Then the ventricular zone gets constant signaling to produce stem cells here, and that stem cell producing area gets thickened. That is called subependymal nodular dysplasia. So this is the area where the neural stem cells generate. So there are excess number of stem cells here, and because of the neuronal migration defects, because of the less number of less amount of reelin, laminin, those kind of signaling proteins, the the neuronal precursor cells will not reach the destined layers, and they end up somewhere in the abnormal positions like this. This is like these ectopic tissues. These form ectopic tissues. These ectopic tissues are called heterotopias. Usually, these heterotopias are the one they create. They trigger seizures. So in this, we can see the colored areas. Colored areas are mostly affected. These colored areas show multifocal. In these colored areas, the brain cells are not properly mature, not properly connected. This is not visible on the MRI, but in the tissue studies, it can be seen. But it's only can be observed in the postmortem brain tissue studies. So you can see these are the areas which so blue colored areas are associated with the social impairment, and yellow colored areas are associated with the communication deficits, and the red color areas are associated with the repetitive behaviors. So these are the abnormalities that are seen in the autistic brain during embryonic development. So it's been since long time people started working on what is the molecular basis of these aberrant development errors, and recent advancements in the next generation sequencing, Illumina and other things, genomic sequences, epigenetic studies. People did some genome-wide association studies from the postmortem brain tissue studies. So very less number of studies have happened in this line. So they have seen a peculiar pattern of gene expressions. One is called, there are three important epigenetic errors that are happening in the autistic brain. One is differential gene expression. Differential means it's a comparison between the normal individual and autistic individual. So they have seen around 584 genes are overexpressed. That means if the particular protein is getting abundantly produced more than the required, and 554 genes underexpressed. That means there is a deficiency of those proteins produced. And differential alternative splicing, this is a peculiar pattern. Alternative splicing mechanisms are normally, this is normal phenomena. Like we have 25,000 genes, but we produce 5 lakh proteins in the body. So this happens with the alternative splicing mechanisms. So I'll explain about alternative splicing mechanism in the next slide. So this mechanism is affected in the autism. This is happening for 833 genes, and this is the pattern is different from the normal individuals. So other pattern is attenuated cortical patterning. There is like how gene there is a usual difference between the frontal lobe and temporal lobe in the gene function of the 523 genes. That is transcription or translation. This there is a normal difference between the frontal and temporal lobe. This is aberrantly this is attenuated in the autistic brain. One of the mechanism is like FDG PET CT, where we can see that the glucose metabolism is affected. That we can be visualized in the PET CTs. So that this is one of the important pattern that is seen in the autistic brain. This is called attenuated cortical patterning. So this is the alternative splicing. So you can see the DNA. Whenever there is a requirement of some proteins, so the particular DNA sequence is selected. So the DNA sequence, so is not, the coding part is not continual. The DNA has coding and non-coding parts scattered like this. Coding parts are called exons, non-coding parts are called introns. So in the RNA also, from the DNA, RNA is transcribed. Then RNA also has coding parts and non-coding parts. These non-coding parts are cut and cleaved or cut, and then these coding parts are joined in a different fashion to produce different proteins. It's like same DNA, same length of DNA could produce 3 different types of proteins. These proteins are called proteins are called isoforms. So this kind of mechanism is aberrantly happening in autism. So if protein A is required, protein B or protein C are formed, and this is happening for around 833 genes. So this is happening because of that brain proteins are abnormally produced. That's why they are unable to like even the neurotransmitters also, like they are proteins. The sensory issues, fine motors, and cognitive ability, many of the mechanisms, and are affected because of this particular mechanism. So, and the question is what is causing these epigenetic errors? Generally, epigenetics means the functions of the gene. So, like how the genes function. So sometimes the epigenetic errors are because of the gene mutations in the human genome itself. Like MECP2 mutation is there in the Rett syndrome. That is an epigenetic driver. Similar people thought similar kind of thing can be identified in the autism. And organizations like Simmons Foundation Autism Research Institute and Simon's Powering Autism did large-scale studies. They identified around 2,000 genes associated with the autism. They're associated, but none of them have more than 1% prevalence in autism. That means this is not, autism is not genetic. Genes are involved, but it is not genetic. The gene structure is normal, but gene functions are affected. That means the gene is abnormally functioning. Why this is abnormally functioning is, it is basically because of the environmental interactions. Here, the environment refers to the gene environment refers to the fetal environment. The fetal environment, there are 2 important things. The one is amniotic fluid, and one is umbilical cord blade. So these are the 2 things that are actually coming into the in the vicinity of the fetus. So from the amniotic fluid, you can have multiple things can be there in the amniotic fluid. Infections may be there, inflammatory molecules are there, or something may be there, because if the uterine environment is not proper, those kind of things can get into the amniotic fluid. And umbilical cord blade that comes from the mother, that actually nourishes the entire fetus for the complete development. So any nutritional deficiencies, hormones, and mother's gut microbiome metabolites, and any antibodies that can travel into the baby. And earlier we used to think that placenta has a very good barrier and it will not allow everything, and it's a controlled environment. But placental barrier also has certain limitations, and many environmental toxins are entering into the fetal circulation. So so to study this one, so not only autism, many conditions are on the rise. So Environmental Protection Agency and Racial Network from the US, it's a government body. They studied the umbilical cord blade toxins in 2007. They only studied in 10 samples probably, what I remember, and they identified around 287 toxic compounds. And out of them, which 237 toxic compounds are neurotoxic. So this is the toxic compounds, and most of them are pesticides, pesticide residues, weedicide residues, industrial toxins, preservatives, and microplastics. So our children are being affected with all these toxic compounds. These toxic compounds generally they directly can have teratogenic effect and induce epigenetic errors. So these toxic compounds. Another thing is in the amniotic fluid. The recent studies shows that in the uterus we usually think that it is sterile. There is no bacteria in it. There is nothing in it. But the recent studies show that amniotic uterus has a peculiar microbiome, which is evolutionarily related to us, and that is the essential microbiome, essential core microbiome that people inherit from the before birth itself. So it is like amniotic fluid has a microbiome. This is the essential microbiome. So these few studies are there on this, but I strongly believe in it. Why? Because in the microbiome science, people believe in high diversity across the world, and people think that children won't have a proper microbiome, but it is not so. So gut bacteria has a role in human health. That means gut bacteria has role in all the biochemical pathways in the human body. If at all any gut bacteria has any impact on the human health, that must be present in the hypothetically healthy newborn. This is the logic behind that there is an existence of microbiome in the womb itself. So this microbiome is the crucial. So another study that makes me feel that this microbiome is important. So we have drawn some hypotheses that this environment toxins and all of these environmental toxins, pesticides, weedicides, industrial toxins entering the fetal circulation. This is not allowing this amniotic fluid microbiome to colonize in the fetal colon, and which is causing the epigenetic errors. So if you prevent these toxic compounds entering the fetal circulation, this microbiome survives in the fetal colon, and it will give a toxin-free environment for the baby and help this baby to develop normally. So this is the working hypothesis we are conducting research in these lines, that environmental toxins preventing this pioneering amniotic fluid microbiome, preventing the amniotic fluid microbiome colonizing in the fetal colon, which is resulting in the epigenetic errors. You can see there is one study. So this is the study that makes me feel that in this study they have taken the gut microbiota, that means the stool from the autistic children. They have transplanted this stool into the pair of germ-free mice, and they have observed their offspring. In their offspring, they have seen a sort of similar epigenetic errors are happening during the fetal development, and they developed a mental illness as well. So the alternative splicing mechanism, differential gene expression patterns, differential metabolomic patterns were similar to human autism. So the gut microbiome the autistic children possess, that itself is a causal factor for many of the epigenetic errors. So this is the hypothesis that human genome of the fetal human genome and the essential microbiome from the amniotic fluid and the environmental factors. These are the three things that govern the entire epigenetic pathways of the human body. This epigenetics is the thing that required for the entire development and running of the entire physiology of the human body. So whenever this essential microbiome and environment factors are disrupted, then epigenetics go deranged. So if it happens before birth, developmental errors occurs. After birth, if this happens, then physiological errors occur. Then physiological errors that turn into pathological conditions like obesity, diabetes, cancer, and cardiovascular disease, Parkinson, Alzheimer's, IBS, and IBD. It's all because of these two factors. So if you focus on these two factors, you can even for the parents or anybody also, the adults also, if somebody focuses on these two factors, then they can have a good health. So, so with this concept, we understood that so there are two solutions to the autism. One is at the individual level. Individual level, so we need to detox the children and do a gut microbiome modulation for their better quality of life. And so for people going for pregnancy, it is applicable for the entire population. But right now, what we are suggesting, if somebody has children with autism and looking for a second child, they can go for first inheritance pattern. They can test their genetics. Parents can test their genetics and the child also and see the inheritance pattern. Then followed by do a toxin burden test, look into the toxin compounds that are accumulated, go for a detox, and if you have some test your microbiome and then modify your microbiome using diet and probiotics, and then go for a pregnancy to give a toxin-free environment for the baby. That will help the baby escape autism. So. So for the children with autism, like if they have if they are comfortable and are having mild symptoms and only if it is only autism symptoms, you can just focus on their diet and probiotics and prebiotics, appropriate probiotics and prebiotics. Like diet, so we should never give them sugar or something like that. Sugar, pesticide containing food, and this junk foods, we should avoid it. Sugar is most dangerous for these kids. And probiotics and prebiotics, you can try this kind of lactobacillus reuteri, which is obtained from the breast milk. A few other lactobacillus strains are there; they're very good. And another probiotic called Clostridium butyricum. We need not worry about the Clostridium species, but Clostridium butyricum is a very good probiotic that is not available in India. Probably one or two companies are there, but one company from Japan is producing it, and it is called as Miarisa. It is a good probiotic so that can help improvement in the brain function as well. And if the children are suffering from multiple immune-related issues, gut symptoms, constipation, diarrhea, colic pain, some sort of eczema and urticaria, and other immune-related disorders, and severely hyper, and many conditions are there. Those children can be tried with gut microbiome transplant. So this is to replace the essential microbiome of the amniotic fluid. If the gut microbiome transplant is done, they can do well. So to replace that amniotic fluid microbiome, so there are three solutions to it. One is poop transplant. We have to take the poop from the healthy child and make it as a solution and give it into the colon from the anal region, from the anus. This is the target area organ where we can directly push in with no much side effects. So healthy amniotic fluid. It is very difficult to execute, or it can even create some problems. So it is not considered amniotic fluid infusion. And the third thing is live biotherapeutics. If we can develop this concoction of the live bacteria from the amniotic fluid, that can become safe and most effective. This may happen in the future. Right now, it is not available. So currently, it is only many people are trying to develop some good bacterial concoction. Like Arizona State University is also working on full spectrum microbiota obtained from the healthy stool. So this kind of efforts can happen in the future. This might take a longer time, maybe a decade or two. So this is at the level of individual, and for the community level, we propose that N-acetylcysteine inclusion in the antenatal program. This takes a longer route for us to make it as a public health policy. But for currently, if people are looking for it, they can go for a detox protocols and they can go for microbiome modulation therapies using diet and probiotics, and they can make sure that they are free from toxic compounds, and provide a toxin-free environment for the baby to develop. Then go for a pregnancy. So that will avoid autism kind of conditions, autism global developmental kind of conditions in the future, in the next babies. So the way we do gut microbiome transplant at Resplice Institute is it is a bit different from others. We collect. So donor is the key in the success of the gut microbiome transplants. So we have a different thought process in the donor criteria. So the entire world takes the adults as donors, and they believe in the high diversity. But we believe that high diversity is not needed, and there is an essential microbiome that is getting inherited from the mother itself in the intrauterine life, and that is the essential microbiome. Every microbe that colonizes after birth is a passive inhabitant, and which is not essential. So to replace, so any treatment should be focused on replacing that essential microbiome. So to avoid the risks of taking adults, we have confined to the children. So because for the adults, we are not sure about their sexual habits, and anything can happen, and they are more exposed to the longer duration of pesticides and other toxic compounds. So then children would be the best donors for as far as my research is concerned. So the first 3 years is the best, but collection of stool from the first 3 years is very difficult. So we extended up to 12 years. 6 to 12, the donors will be recruited. So they will be screened with a battery of tests that are suggested by International Stool Banking. Then we also do a food intolerance test, which is a quality indicator for the microbiome. So that test also we do. Then we recruit the children, and then collection process also has a specific method where we need to maintain some aseptic procedures. Like even the collection also should not happen in the restrooms, because the restrooms have high load of bacteria that can contaminate the stools. So it has to be done in a specific dedicated room. So with sterile drapes placed, it should be collected in a sterile container. We use gamma sterilized containers for that. Then that stool is transported in 4 degrees centigrade to the clinic, and then it is by using homogenization and refrigerated centrifuges, we produce different types of solutions, process it, and it will be infused into the colon on a daily basis for at least 2 to 3 months. Then, so if we are doing such kind of thing for a longer duration, then the microbiome transformation can occur. So that will change that will improve their microbiome, immune system, neurotransmitter balance, hormones, enzymes, deficiencies, and many things. So the food intolerance test, we were talking about the food intolerance in the previous slide also. So this is the gut lining. In this gut line, over this gut lining, there is bacteria here. This is called gut flora, gut microbiome. Just beneath that gut lining, 80% of immune cells stay. So these two immune cells and gut bacteria have a molecular crosstalk. So, and they act as a gatekeepers for the system. So anything that we consume enter through the gut lining. Before entering into our system, it is being checked by these gut bacteria and immune cells. If you have a human compatible and healthy microbiome, this will not report our food as antigen to the immune cells. That means if you have a good bacteria here, then there is no food allergy or food intolerance. If you have a bad bacteria, then it will automatically trigger immune system. So then there is food allergies and food intolerance. So this is the impact we study. That so why we are not relying on just testing this bacteria. So we do test that bacteria, but so the problem here is like the 16S rRNA sequencing or shotgun metagenomics. They just give a classification of the bacteria, but impact of the bacteria is not seen in that. So if some pathogenic bacteria is there, if some regular commensal is there, but that is infected with some bacteriophage which contains virulence gene that can turn into the pathogenic bacteria, those kind of information is not available in the regular microbiome test. So, so we are also studying this impact of the microbiome also. So this is like if somebody doesn't have a good bacteria here, good microbiome, they will develop food intolerance or food allergy. So before going for the treatment also, one can test food intolerance and see whether they will be a responder to the treatment or not. If somebody has a good bacteria already, they will not respond. By giving good bacteria, they are not going to get any sort of benefit. So they need not go undergo this kind of treatment. If somebody has a bad bacteria, they will have many food intolerances, food allergies, immune-related issues. They can go for this treatment and they can respond better to this treatment. So what are the benefits that can be expected after this? So once you achieve that microbial transformation, so it starts training our immune system. So we all talk about yeast overgrowth and other allergies, intolerances, and urticaria patterns and other immune-related disorders. We can all see this immune system relation dysregulated patterns in the autism. All this can settle with good number of sessions of gut microbiome transplant, and gut symptoms are sure to improve, like constipation, diarrhea, colic pain, and digestion issues, non-specific colitis, anal fissures, etc. All these gut symptoms can improve, and this also brings neurotransmitter balance. With the neurotransmitter balance, you can their irritability and sleep disturbances, mood disturbances, and sensory issues, fine motors, learning ability, cognitive ability, all these things will improve. And gut microbiome is known for digestion and absorption. So vitamins B and K and many other things can improve. And hormones and enzymes also, they can come into the balance. These are the benefits expected. And so when coming to the pathology of autism, the mechanism that that were related to the autism, when we if we do a gut microbiome transplant, what are the mechanisms that show some effect? So in the neuronal migration kind of things happen inside the womb itself, and those kind of things may not have effect after birth. So gut microbiome transplant may not be showing any effect on the neuronal migration, but neuronal differentiation to some extent in the first few years, like up to 4, 5 years, 6 years, when there is a good neuroplasticity, at that point of time, if gut microbiome can signal, produce signaling molecules to different for the differentiation, and microglial dysregulation also can be corrected with a gut microbiome transplant. The immune cells get trained, microglia are the immune cells, and they can cut the excess number of synapses and improve the synaptic pruning and functional circuitry formation also. With this, the cognitive abilities improve, learning capabilities improve, sensory issues improve, and fine motors improve. These are the mechanisms. And axillary neurogenesis that we talked about, that ventricular zone thickening, that cannot have any sort of improvement. So these are the and when we look into the gross pathology abnormalities. So the subependymal nodular dysplasia heterotopia that we discussed earlier. So I'll show you the diagram now. So though these two may not have any effect, multifocal dysplasia might have some benefit, because this gut microbiome keeps giving signaling molecules that differentiate the neurons, that help the neuronal maturation. So then multifocal dysplasias might have some benefit. It is also in the active neuroplasticity. So I'll show you the previous picture. So this is the heterotopia. This already migrated to a different place, and it is in the soft tissue. This cannot go to the destined place again. And subependymal nodular dysplasia. This is the thickening of the ventricular nerve. This also cannot be repaired. And so this is the multifocal dysplasias. This might have benefit to some extent, but complete development may not be there because the mechanisms are happened in the inside womb itself. So another thing is we also started one more program called SmartLand. This is for maternal environment modification before pregnancy for autism prevention. Autism is a preventable condition. So if we cut down all toxic compounds entering into the fetal circulation, that will give a toxin-free environment for the baby. Then they can avoid all these epigenetic errors and they can develop into normal individuals. So I believe that 99% of today's children with autism were supposed to be born normal, but unfortunately they lost their life because of environmental toxins. So if we cut down that one and improve the functional, improve microbiome of the mother, and any antibodies are there from the mother, or any functional deficiencies or other things, hormonal problems. If we correct all those kind of maternal environment issues, the child will definitely have a healthier development. So this is our belief, and we started doing it actively now. So if somebody is seeking for a second child after having a first child, autism in the first child, they can look for this one. So this is the so I've compiled research articles and I've created a QR code. Please, you can scan this QR code for all the research articles. And so I've given the number of our coordinator. You can note down this number and you can coordinate for the consultations. And lastly, regulatory aspects of the gut microbiome transplant. FMT is called as fecal microbiota transplant or gut microbiome transplant. It is US FDA approved for Clostridium difficile and ulcerative colitis. These are the two conditions it is approved. It is not yet approved for the autism, but its efficacy is observed by many researchers since long time. Even Arizona State University from the in USA, they published a study in 2019, and they have done a phase 1 study, and now they are into the phase 2 study. They have got a good result in the phase 1 study. Dr. James B. Adams is the principal investigator of that. So that study was the inspiration for my studies. We have registered one study in the 2022. So our study is in the first phase. And it is being around more than 400 clinical trials are being done for various disorders, and around 10 clinical trials are there across the globe in the autism. So this is the take-home message. So autism is a preventable condition. So people who are looking for a child, they can go for a detox programs and improve their gut health and improve other conditions that can thyroid and other hormones and immune-related issues. They can solve all those problems and go for a pregnancy, so that there is no autism or global delay or any unwanted conditions in these children. And autism, for the affected children, they can be improved to a great extent with microbiome modulation. Even if somebody is going for a stem cell therapy, exosome therapy, or functional medicine approaches, they do better. All those treatments get more effective after microbiome modulation. So microbiome and environmental toxins are the two major modifiable mechanisms, modifiable factors that needs to be addressed to improve the children with autism, and also for the providing toxin-free environment for the babies to develop normally. Thank you, and thanks for joining the webinar. Any questions?
Hello, sir.
Hello.
Huh. Sir, can you just guide us whether GMT we have to follow some dietary patterns every day,
Yes,
and
yes.
and with that we have to follow some antibiotics to flush out the micro.
Yeah, so this was an earlier thought process that vancomycin was used to be given. In my trial also vancomycin is there.
What is that,
But
sir? Micro?
vancomycin.
What is that, sir?
Vancomycin is a antibiotic. So before gut microbiome transplant, people give it for 2 weeks. So that existing microbiome gets suppressed. The new microbiome, it will improve the new microbiome chances to engraft. This is the idea behind vancomycin. But
Vancomycin. What is the
vancomycin,
spelling? Can you please
V
give
A
us?
V A
Just a
N C
minute.
O, yeah,
V A
VANCO
N C O.
MYCIN.
This is the antibiotic.
This is the antibiotic the usually people use before gut microbiome transplant. But so what we have observed, even if you give vancomycin, so if you look at vancomycin resistant Clostridium difficile infection, the treatment option is gut microbiome transplant, that is fecal microbiota transplant. That means gut bacteria has better antibiotics than vancomycin.
Hmm.
So, there is no need of vancomycin before giving fecal transplant because gut bacteria produce more precise and better antibiotics than vancomycin, and they possess a mechanism called quorum sensing. You can read about quorum sensing. They can identify the genomes of the other bacteria, and they can directly target their genomes and for their survival. So, whichever
Can
the
you spell
bacteria,
it?
quorum sensing, QUORUM,
Q O Q
O
U O.
RUM,
Hmm.
sensing, SENSING.
Sensing. And just what are the chances if we do the GMT and we follow all the diets and all the my antibiotic processes
Yes.
as per the prescription given by the
Mm.
doctor? What are the
Mm.
chances that the child with a severe autism will recover?
Severe autism, like if you look at the like if the child is severe, if somebody is completely non-verbal, that means the speech areas and language areas are not developed during brain development. So they have less chance of getting speech and language. But if the areas which are developed because of the epigenetic errors, they are dysfunctional. So you can improve, you can stimulate those areas. So when you do a gut microbiome transplant, the developed areas which are dysfunctional can be activated. That will improve their quality of life. So this is the benefit they can have. If somebody is severe, also they can improve to a certain extent.
So, can you just again give the idea which are the parts of the brain is hampered in the
Yes.
autism? You have
Okay.
already showed that slide, but could you please explain it again? And
Yeah.
that too for the speech and cognition.
Sure, sure. Cognitive function is the entire brain's function. Cognitive function. It is like diffuse brain function. For the speech, there are areas called Broca's area, Wernicke's area, and language also has some certain areas. Language areas, like they are linked to the auditory cortex. Then, the whatever the information they perceive, their comprehension, vocabulary, and everything is processed in the language areas. The speech is being produced in Broca's area and Wernicke's area. So
Brokers and
yeah, so those are the areas of the brain. they produce speech. If those areas are not properly developed, they cannot get a speech also. So even after gut migraine transplant, so so for some children who have some speech, there will be improvement in the speech.
Okay, and sir, if a child was having language, having speech, but by some reason,
Mm.
maybe by some what we can say, by some reason, he regressed. Then,
Regressed. Yeah. So there is, I'll tell you. So I'll tell you about the regression patterns. We all have like some concept of regression. Like we all think that because of that regression, we all think that autism is occurring after birth, but it is not so. Autism is occurring inside the womb. But what happens? They inherit a susceptible microbiome. So the microbiome, this gut bacteria has a role in neurotransmitter balance. So when this gut bacteria is exposed to certain things like vaccines and antibiotics in the first few years of life, then that can disrupt the bacteria further. When that bacteria is disrupted, the neurotransmitter balance is further disrupted, and they lose the skills. This is regression actually. So, but before regression also, if you see that those children, they have such kind of patterns, autism patterns will be there. So they regress further. So in our clinical trial also we observed that if somebody has gut symptoms, if somebody has documented food intolerance and blood test, and if somebody is having regression, so these three criteria of children improve. So after gut microbiome transplant, whatever the skills that they lost before regression, they are recovered.
oh, nice, nice.
This was the observation we have seen. This is the three criteria, like somebody has gut symptoms, somebody has food intolerance positive, and regression. So these three criteria, if somebody meets this criteria, they can look for the gut microbiome transplant.
Huh. Sir, can you guide us on the diet pattern then?
Diet is like you can follow Mediterranean diet, so any diet you can. It's like I'll tell you. So every human being is having a instinct, and they have a food apparatus in their body. This is developed in evolution. If you are put in an island which is well flourished and you don't have any tools to kill any animal, if you don't have a tools to cook, you don't have any kitchen or anything, whatever you can eat from that island is your food. That means you can eat all the fruits and vegetables, and even the grains are not our food. We adapted in the during for the survival process. Meat is also not our food. People claim that we have canines, and these canines are there for the most of the monkeys also. This is nut-eating canines, and the other this is not similar to the carnivorous canines. And people say that early humans only survived on hunting. It is not. It's a survival mechanism actually. So they ate this like burnt meat in the wildfires, and they understood that fire can make it edible for us. Then they adapted meat as food. And grains also it is like that. They cook it and ate it. And anything that you can eat in the raw form, and you get instinct to eat, that is our food. So with that principle, you have to see. So whether that can be that is our food or not, that will show impact on the children's microbiome. So so sugar is very dangerous. Sugar, when you we give processed sugars or jaggery or anything, they definitely get hyper. So it is actually it makes their gut breeding ground for the bad bacteria. And pesticides and weedicides and all these things are very dangerous for the microbiome. It is like a trigger for further disruption. So I lost your voice. So you are on mute. Can you do something? Sorry, you are not audible.
And now, sir,
Yes, yes, you are audible.
ha.
Please tell me.
Sir, I wanted your help. If I share one case with you, could you please
Okay.
help me out with that?
Yes, yes. Yeah, please tell me.
Sir, I know one child who is maybe moderate to severe autistic,
Okay.
and she was speaking little words behind us, and now she
Okay.
regretted.
Okay.
We had done stem cell therapy on her for 3 times.
Okay.
Huh. So she is improving. Her understanding is improved, but
Okay.
she is not verbal.
Okay.
So
So,
could you give us the
okay. So you can check these three patterns. Like, does the child have any constipation issues, or colic pain, or any immune related issues? And
pardon,
you are
constipation? First of all,
yes,
constipation. Then
constipation or diarrhea patterns, or poor stool quality, or any colic pain, or other patterns like allergy patterns. You can see all these things, and if you can check food allergy and food intolerance test, that will tell about their response. Like, you can actually find out who is the responder before going for the treatment itself.
okay. So what are the chances if we go with the GMT test and we follow all the diet patterns, all the antibiotics? What are the chances that the regression, what she was having the speech earlier, will get again?
Yeah. So it is if somebody has a bad bacteria and you are giving good bacteria, then they will definitely improve. It is like, if you are having iron deficiency and you are given iron injection, you will improve, right? It is like that. If you can identify somebody having bad gut bacteria and change it, automatically they will improve. How to see that they have bad bacteria or not? So this is like, if the gut bacteria was the reason for the regression, if they have a bad diet and bad something that disrupted the microbiome, then that created a neurotransmitter imbalance and then regressed. Then you replace that gut microbiome. Automatically, biochemical balance will be reclaimed. Will come into the balance again, and they start developing the skills that they lost.
Okay, and we means whatever loss she'll gain, but
Yes.
whatever she has to gain, like language,
So,
if she
yes,
has.
that I have told you, right. The multifocal dysplasias also have some effect, but that is a slow effect.
Okay, okay.
So, but whatever the last skills are there, they will recover in first 2 to 3 months itself.
But sir, as like I wanted to ask you this, whether we are following everything, but if she is very hyper, if she see biscuits, if she see chocolate, she'll grab it like anything and she'll start eating. So,
Okay.
would you recommend that some hyperactivity curbing medicine should be given in this stage so
No.
that she will not.
If you are not going for gut microbiome transplant, then the child might need. But so once you give a chocolate or anything, that will create a craving kind of thing on a continual basis. But the only thing that you can is best to do is stop them from eating. If you do it strictly, then if they don't have that component in their body, that will not create craving. So once there is no craving for it, they will not look for it. So
But
at least,
what happened now for doing that also? We are socialized.
so that is our that depends on the parents' commitment. So if you want
Ah,
them,
but
yeah, if you want them to have zero screen time, if you don't want them to eat chocolates or junk food, it is the parents' commitment. Like, if you feed sugar to them, then that sugar will create craving to them. So that will keep continuing. So that we need to keep an abstinence from that. We have to be strict on that, and we should not give any sugars or junk food to them. So that is in the hands of the parents.
sir, how long this diet should be followed?
So this is for the if you are not going for gut microbiome transplant, this has to be followed for a longer period. At least 2 to 3 years, we follow that. Then their microbiome improves.
The gut
So then
microbiome transplant is the process, right?
it's a process. Yes.
So what is that process? Could you please again
So
explain it
you
in
need to
simple words?
yeah, you need to identify a healthy donor, healthy child, and you need to do a battery of tests on them, and inquire about their entire family history and other things. So then you collect their stools on a daily basis and prepare a solution. Then every day solution will be tested. Then that solution has to be infused into the affected child's colon. A tube is implanted from the anus to the colon, and it is injected into that. So
And
this is
where
the
this
process.
process can where
So right
how
now,
can we do?
yeah. So right now, very few places in the world are doing it. Some a few hospitals in Australia are doing, and one hospital in Mexico is doing, and in India, one doctor in AIMS and one doctor in some private hospital is doing, and in our research in our institute also we are doing under research now. Uh, so uh, so people do it by themselves. Many of the people across the world they do it by themselves. Yeah. So it's a do-it-yourself procedure as well. People can learn and do
It's
it at
very
home.
difficult to do by myself. Ca-
So it
In
can be
India,
trained.
we
Simple
huh?
procedure. It's a simple procedure. One can get trained and they can do it by themselves. It's not a big. The only key factor is the donor.
Where
If
can
you
we
have a
do?
donor, if you have a donor, proper donor at home, people can do at home. We have trained many parents, so they can they did at home. They are doing at home also.
Okay.
Yeah. Mm.
Can I go ahead with my question?
Yeah. Please go ahead.
Yeah. So thank you so much for the presentation. So you talked about the diet, right? So now it is more like we need to give like protein and everything. So what is your take on that, doctor? So whatever that you mentioned, I don't think so. There is much of a protein option. So what is
Yeah.
your take on that?
So for the protein, okay. So we have so if you check for the food intolerance, also most of them get food intolerance for the nuts as well. But you can so you can give egg and many other are there. You do a test for food intolerance test. So in that food intolerance, food intolerance and food allergy, you will find out so what kind of foods they are showing some inflammatory process. If some like if you don't have any inflammatory process going on for egg or something like that, you can use those kind of foods for protein mainly.
Food tolerance test, and we found out that he has issue with gluten,
Mm.
just gluten. So can you suggest a dietary plan except gluten, like just an example of dietary, how the entire day, what all the items that we can give?
So okay, he has only allergic to gluten.
Yeah, he's allergic to gluten.
Then you can just go for the BFC of diet.
So what is your take on vegetable juices, doctor? Is it good
Vegetable
for
juices,
the?
yeah, it's good. It's good. It's good, but you need to see that it should be organic.
Okay, okay.
Vegetable juices, like that's what I was saying. No? Vegetables and fruits are the important things that will transform the gut microbiome. You can look into the what is Mediterranean diet. So that's a good diet plans. You can use that. It's like anything you can eat in raw form is good actually.
Yeah. So whenever we give gluten, right? So he has eczema. It is getting aggravated. Is
So
eczema
then,
related to gut issues?
yes. So it's basically the food allergies, food intolerance, everything starts in the gut.
Okay, okay.
Yeah, you work on the gut health, then they improve. Allergic patterns improve.
Okay, doctor. Thank you so much.
Thank you. Any other questions?
Yes, sir. Sir,
Yes.
for protein, can we go for egg?
Yes, yes, you can go for, but you need to see whether they are allergic to it or not.
No, I don't think she is allergic because she every day eats egg.
You, yeah. So if you can look, you can objectively test it. Probably food allergy and food intolerance test will be available in every city. You can go for that test and see whether is it creating any inflammatory process in their body. If it is not creating inflammatory process, then you can give it.
Yes. Yes.
Yeah. Doctor, I have a question. Can I go ahead?
Yes, please.
Yes. See, actually, my son is 11 years old and he is on the milder side as such in academics and all, but
Okay.
very much autistic. Okay, the autistic part is there. In fact, I had come for one consultation also once I had spoken to you in Hyderabad, but later I
Okay.
did not go ahead with the FMT
Okay.
thing because just I was extremely scared of that at
Okay.
that time because of multiple
Okay.
behavioral issues.
Okay.
Now my only question is like if we are not going ahead with the FMT, okay, because I'm little because it's related to a donor. I like that
Yes.
entire. I mean, in theory, it is very good. Okay, the approach is good, but when it
Okay.
comes to my son, I'm little scared, little hesitant. That's the only thing.
Yes.
But if we don't go ahead with it, okay, is there any other thing that can be done to clean his uh, as in to clean the gut health or some probiotics, customized probiotics? Can do you consult for all of that?
Actually, you can do that. Actually, you just focus on bowel cleanse. You can try certain probiotics. There are two. So in India, if you can, India, you can see some probiotics like Protect This that comes from Reddy's Labs. So they produce a live bacteria called Lactobacillus reuteri. So you can, and another probiotic called VSL3. So that comes in capsule forms. If they maintain a proper cold chain, that also works. These two things are there, good probiotics. And one probiotic from Japan, it comes with a name called Niarisan. It's Clostridium lubricum. So these three probiotics have seen very good improvements in the children. So that you can try. So if the child is uh, having allergic patterns and gut symptoms, then you can look for the gut microbiome transplant. So if the child is not having problem and milder at mild only if the child is overall comfortable, then you can just focus on the diet and probiotics.
Diet and probiotics, but you don't consult for that, is it? Customized, you don't develop for the child, is it?
So
In your unit,
so we can so we have consultants for that. I don't do that, but we have consultants for that.
okay, I
So
understand. Okay.
we have some functional medicine doctors and dietitians. They do that.
Okay, because I'm already giving this Ruteri. I just read through that. Okay, BioGaia, there was one
Yeah,
company.
yes, yes.
Through that, okay, I started giving this.
That is very good. You can give. So you can actually prepare curd with these probiotics. Actually, they are live bacteria. They are better than spore forms. So most of the other probiotics that are available are spore forms. Though it is called colony forming units, they will not have like longer effect. They are very short term benefit will be there. So if you have if you are looking at transforming your microbiome, you can use these probiotics and make some curd. So if you curd rice also you can prepare. So that
But I had stopped curd and milk long time. First thing I stopped was that.
yeah.
Oh,
So because of the casein, people stop it. But if you are making it yogurt or curd, you can give it. What I feel, because the bacteria is important. So if the bacteria is modified, then if the bacteria is modified properly, then automatically that will reduce the inflammatory process to the casein.
okay. I got it, doctor. And one more thing, okay, what is you said spore bacteria, spore probiotics are not great, but microbiome labs, that spore is it's very popular. I think developed by microbiome labs in the US.
Okay.
None of the spore are good, is it? Because
So
I
spore forms
heard
are very short. Yeah, they are short acting kind of thing. So they don't colonize. They don't transform the this thing. So all the spore probiotics are like I can explain you in a simple understanding form. Like the spore probiotics are like bullet shots, and gut microbiome transplant is like a nuclear explosion. This is the comparison between these two. It's a simple logic.
okay, but if we give, there will be no harm, right? Because I've not started. Um,
So some probiotics are not that good. Like I don't know,
okay,
maybe spore probiotics. So I've seen multiple problems with these children when we are giving spore probiotics.
okay,
That was the reason I generally stay away from probiotics, spore forms for these children, especially spore. Only few probiotics I prefer. That will high biotherapeutics I prefer. So other things I try to be away from it.
okay. Thank you.
Doctor, I have one more question. So, what is your take on prebiotic supplements like inulin?
Prebiotics is good, but in some cases some parents reported that they have seen some sort of behavior patterns or something like that. But otherwise, when I gave to few children, I didn't see much problems. So I feel it is good. We can try.
Uh, can you can you tell the can you tell the early signs of autism to identify, and what is the best stage to start GMT, and what is the upper limit to start, and what are
So
the
yes, yes, sir.
yeah.
So this is like we need to identify the children in the first few years. Like so, when the neuroplasticity is intact, neuroplasticity is like brain's capacity to rewiring is good in first 3 years itself. It is also good in 3 to 6 also. The children should be like if the children are identified at the age of 2. So we have seen best benefit in the younger children. So with the age also, first 6 years is the best time. If they undergo treatment at the age of 2 or 3, they show much better improvements. So it can help in at any age, but the first 6 years is the best time to do it because people do it for Parkinson's and Alzheimer's and still get benefit out of it. Generally Parkinson's, Alzheimer's, all these things happen in the old age. They also get benefit out of gut microbiome transplant. For autistic children, we are trying to rewire their brain connections. That is good in active neuroplasticity years. So that is first 6 years is the best time.
Okay, okay, understood. Okay, thank you.
Thanks.
Hello,
Hello. Yeah. Please tell me.
sir. I'm from Pune,
Okay.
and my son, 13-year-old, is
Okay.
autistic. So now, how to identify which treatment will help him? He's a non-verbal.
Okay. Okay. How to identify which
Means
is
which
best treatment?
with whether the only the probiotic will help him, or the GMT what you explain
So,
that
does
will
the
help
child
him.
has any gut symptoms? Constipation, diarrhea, or colic patterns, or abdominal pain?
No, no.
So all these things are not there. Hmm?
Yeah.
So is child is suffering from any allergic patterns?
No,
Any
not
allergies are there?
not as such.
Except for autism, is he suffering with any other condition?
No,
No.
no.
Is he regressing with simple triggers also? Is he
Yeah, sir.
losing skills?
If we don't give him what he wants, then he becomes hyper and he starts hitting
Yeah.
us.
So those kind
That
of
is the
tantrums.
thing. And nowadays, like he's too much. We had kept him in residential school, and 1 week
Okay.
back we got him back, and
Okay.
we just wanted to see whether he's improved there or no. But the thing is same. He's the same. If we don't give him what he wants, then he becomes hyper and he starts hitting me, especially me, not his
Okay.
father.
Okay.
So it's very
So
difficult to handle him.
Just for a
okay.
few, maybe half an hour in a day. Otherwise, he's happy, but there is a mood swing.
So mood swings will be there for these kids for definitely because they have a different neurotransmitter balance. So for that you need to see like if there are no gut symptoms and if you don't have any documented food allergies or intolerances and there are no regress in balance, these kind of tantrums are there and rest everything is fine. You just focus on their diet first. One is you focus on their diet, what kind of diet is triggering such kind of behavior you see. That is one thing, and look for these occupational therapy kind of things, and you can keep bowel cleanse programs also. That also might help. These are a few things I can say. If you are looking for some medications also, you can try this. You can see a pediatric neurophysician and take those medications like Cognicare or some Risperidone. All these things are there, but they have long-term side effects. So I would rather say you rely on these occupational therapy kind of things.
Okay. But probiotic things can help or
Probiotics. So if the child doesn't have any gut symptoms and other things, and
hmm.
you give probiotics, sometimes it can backfire.
Then it is do you advise we get his stool tested maybe?
It's not stool. You have to test the blood test. If you do
Blood
a
test.
stool test for microbiome,
Hmm.
generally inconclusive. So because it gives only a bacterial classification, and
Hmm.
it doesn't give any information on the pathogenicity of the bacteria. So,
Hmm.
but you can do a food intolerance test, which is a quality indicator kind of thing. So, what we have observed.
Hmm.
So, when we have taken children with food intolerance positive on the blood test
Hmm.
and do the gut microbiome transplant, we see a change in the reports in the subsequent. That if somebody has some 40, 50 intolerances for foods and
Hmm.
they go less than 10 over a period of 6 months.
So I think you advise the food tolerance test, maybe the.
Yes, food intolerance. You do a food intolerance test. Based on that, you can do some kind of. You can try the treatments.
Okay, okay, sir. Thank you, sir.
Thank you. Any other questions?
Sir?
Yeah, please tell.
Sir, can you please give in chat some prebiotics and probiotics which can be started?
Probiotics. So
Yeah,
yeah,
prebiotics and
probiotics.
probiotics for the
Yeah, you
severe
can do
case
one
of
thing.
autism.
Yeah, so you can do one thing. There are two probiotics that are available in India. They are very good. One is it comes with a name called Protectis.
Pro?
Protectis.
Protectis.
PROTEC. Protectis. The bacteria inside it is Lactobacillus ruteri.
Lactobacillus.
Ruteri. Another thing is called VSL3.
VSM3.
VSL3.
Okay. Can you spell
Okay.
in the chat, sir? In call message. Can you give in the in call message?
VSL3 and Protectis.
Protectix.
Protectis and VSL3.
Okay. These are probiotics.
These are two probiotics that are good in this one. So you can actually prepare some
How to
sort of
take it,
yogurt
sir?
also with this. Okay. Yeah. Pardon.
How to take this?
Yeah. So for protect this, no, you can take 5 6 drops in the 5 ml of water and drink it. And VSL3 comes in a capsule form. You can take directly capsule, but so I would prefer a different method. So what you can do is like you can take a clay pot and put some cooked rice in it. pour some water, put some milk in it, and open the capsule and put that powder. And very next day morning, you will see a very good quality curd rice. That you can give it as a breakfast. So
Okay.
protective also you can do that.
Okay, okay, yes, yes.
So this method will it is more effective than taking capsule and that drops,
Hmm.
because the bacteria multiply and you are getting it in a very greater number. So even the gastric acid kills and other pancreatic enzymes kill also. You have still some bacteria left to colonize the food.
VSL3 should be added in this milk,
Yes,
and
yes.
that has to be kept overnight,
Overnight, next day morning you
and
can
then
eat it as
mix
a
it
breakfast.
in rice.
Yes,
Okay, VSL3 probiotics. It's in a capsule format, right?
yes.
Okay, every day, one time.
Yeah, every day. Yeah, so you can put it in the night. You can take a clay pot and put some rice and water in it, and little bit some milk in it, and that powder in the VSL3 capsule, and you can use that as a the very next day morning. You can see good quality curd rice. That you can take it as a breakfast. So this
Yes.
can be done on a daily basis and every day.
But sir, we as some dietitian said that milk products should be avoided. Then this
So, but
curd
for
should
this
be.
is okay. For doing this one, this is okay. Or otherwise, you can do one thing. So, I've seen curdling with coconut milk. So you can use coconut milk and try. If it is giving a curd rice kind of thing, then you can use that also. If there is no option, you can use it this milk. If they're allergic to it, then you can stay away. But most of the people will most of them have, even though have they have inflammatory process with the intestine, it is subclinical level. So in those cases, the replacing bacteria will be preferred over this inflammatory process. If you get a good bacteria, this inflammatory process will reduce over time.
Okay, and this bio protective drops that has to be taken once in a month, once in a day.
Yes, once in a day.
Okay, morning time or evening.
Before bedtime, you can give.
Okay, in the water.
Yes,
Okay, and the diet should be followed strictly along with this.
yes,
Okay, and sir, as you told me, GMT can be done at home, but it is very struggling to get a donor.
yes,
So
definitely.
how would
Because
you guide in this?
so we will help you in so if you have some donors on your mind, if you have if the child has any cousins or siblings, we can test them and find out whether they are suitable, and then you can get trained how to collect the stools and how to prepare the solution and how to
So
give it
how
as.
to get a help from you on this? I want to go for this. So
So we can connect one to one, and then we can figure it out how if you have children, other children at home, I'll help you on that how to do it,
no, I don't have any
because
other
it's a
children.
long process. No? And we're going to be explaining this half the time.
So your number? Okay.
So we'll message that number in sharing this. So I'll share the slides also. That also coordinate number is there.
Yes, please share the call. This is number no 92810.
Yes, yes.
I'll connect with this
Yeah.
after this
So,
call. Yes.
and that slide has QR code. So please scan that QR code and read all the articles. They're very good articles, and that will help you in selecting the treatments for your children. So understanding autism and actual pathology of the autism, that will make you so that will be clear your mind, and that will help you in selecting the treatments. So
Yes.
what is right treatment, what is wrong treatment, so what is relevant and what is irrelevant for autism, you will be able to understand. So those are the research articles. So though they are technically too technical, but still you can read abstracts at least.
Yes, sir. Yes, that is
Very much
that is
important.
given in a WhatsApp group, right?
So that QR code is there. If you can scan the QR code, automatically you can download all the articles. So I've given around 30 to 40 articles in that. All are very much relevant. So that will tell you about entire thing about the autism.
Yes, sir.
Any other questions, please? Okay. Thanks for joining the webinar. Good luck to all. Thank you. Thank you very much.